We'll be doing a presentation on chromosome shattering, a look at constitutional chromothripsis in patients exhibiting multiple copy number changes and complex structural rearrangements revealed by S&P microarray. First, I'll go over a short overview. I will be explaining what defines a rearrangement as complex, what chromothripsis is, and a little bit of background of how and when it was discovered, a few hypothesized molecular causes, along with some devastating results of chromothripsis. I will be explaining or comparing the benefits and limitations of G-band chromosome analysis versus microarray analysis, and how it may be best to have both analysis to fully understand these complex rearrangements. I will follow up with patient examples exhibiting constitutional chromothripsis. Then I'll conclude and give a few acknowledgments. So as we all know, structural variations impact a major part of the human genome. These variations or rearrangements can lead to congenital disease and aid in cancer development. Rearrangements are defined as complex when there are at least 3 or more breakpoints involving 2 or more chromosomes. I would like to point out that the rearrangements involved in chromothripsis are not your typical structural rearrangements. These rearrangements can be so complex that the banding pattern is often not easily determined. So what exactly is chromothripsis? If we break the word down, we have chromosome, that is referring to the chromosome. And then thripsis in Greek translates into shattering. So you put it together and you have chromosome shattering. Chromothripsis is characterized as a catastrophic event in the cell that shatters 1 or more chromosomes. The chromosomes are then put back together in a haphazard way into derivative chromosomes. During this event, hundreds of thousands of genetic material can be blown apart and stitched back together, causing highly complex structural rearrangements. This event may involve multiple chromosomes, but also may only be multiple events on a single chromosome. So let's imagine that this is a normal chromosome with the sequences going in the right order and orientation. An event comes along and shatters these pieces and puts them back together in the incorrect order and orientation. You notice some of the sequences are upside down and backwards. The derivative chromosome is a lot shorter than the original chromosome because obviously some of the genetic material is not reincorporated into the derivative chromosome. This is just another image of how this event or this catastrophic shattering could occur on a localized region of a chromosome. So you have the same shattering event put back together. One portion of the chromosome is left undamaged and another localized region is actually damaged. It was originally thought that most complex rearrangements or mutations came about accumulated over time, basically accumulating progressively and sequentially of one another over many cell cycles. However, chromothripsis is the new model where it is thought a single explosion gives rise to these complex rearrangements. Chromothripsis is a recently described phenomenon, first seen in cancer discoveries in 2011. It was discovered while sequencing the genome of CLL, and although right now there's more knowledge about somatic cell chromothripsis, there is evidence in the literature that germline events may be more prevalent than anticipated. So my patient examples that I'm going to follow up with show the complexity of constitutional chromothripsis in shattering events of the germline. So, as you know, when this event occurs in somatic cells, it gives rise to cancer. When it arises in somatic cells, it gives rise to cancer. When it occurs in germ cells, it gives rise to congenital disease. Chromothripsis can lead to a lot of dysregulated gene expression. This is due to the fact that copy number changes, which can cause deletion of tumor suppressor genes or even application of genes promoting uncontrollable cellular processes. Depending on which chromosome or chromosomes are involved, different cancers and congenital diseases are presented. So the mechanism or mechanisms that initiate catastrophic chromothripsis are not known, but it has been hypothesized that advancing paternal age is a contributing factor. A few repair mechanisms involved would be non-homologous end joining, or less frequently, microhomology-mediated end joining. A few other possible causes would be radiation damage, where free radicals can induce DNA breakage, as well as telomeric crisis, which happens where if telomeres get worn away, this can cause genomic instability. There may be some proliferative advantage to the cell to over-replicate these chromosomes, and there may also be some type of defect in the DNA polymerase. So the incidence of chromothripsis in cancer is about 2 to 3% of all cancers. It's more prevalent in bone cancers, being about 25%. There has been studies followed up to confirm that chromothripsis can lead to colorectal cancer, AML, and multiple myeloma. The incidence of congenital chromothripsis, however, is a lot less clear. This is mainly due to the fact that systematic screening has not been performed in a large group of people quite yet. So fortunately, we can use G-banded chromosome analysis to observe these complex rearrangements. Chromosome analysis is a whole genome methodology where we are able to visualize structural rearrangements even if the composition is not clear. However, the phenomenon of constitutional chromothripsis is not often appreciated by routine chromosome analysis. This is mainly due to the fact that copy number changes are often submicroscopic, and the rearranged segments may not resemble known or recognizable banding pattern. This means the resolution can be a problem. Even when you have high resolution chromosomes, the smallest visible band can still have up to 2 to 5 million base pairs within it. So depending on which chromosomes are involved, this may result in an oversimplification of the karyotype. Microarray, however, is also a whole genome methodology where we are able to use it at a much higher resolution. Microarray can reveal complexity that's not appreciated by chromosome analysis. However, the structure can only be inferred, if at all, and balanced rearrangements cannot be detected. So the images on the left are from a published case from our lab where the array showed a simple unbalanced derivative 12 from a 312 translocation. And once we did the chromosome analysis, we found a secondary rearrangement between chromosomes 3 and 11 that was balanced by the array. So this case, along with the cases that I will present, are all examples of where no one test is sufficient. So our first patient had chromosomes previously done at 6 months of age due to tracheomalacia. The patient was referred from microarray to clarify this previous chromosome result. The patient was reported to have a deleted 3q and a translocation between chromosome 6 and chromosome 9. Array showed a 7 megabase deletion and the Q arm of 3 near the proximal breakpoint noted in the chromosome analysis. And I'd like to point out the 3 tracks up here are the S&P pearls on the chromosome. And a normal array will have 3 allele tracks, and a deletion will show a loss of an allele track. You can see this right here. Array was balanced, 6 was balanced by the array analysis, but the 9, however, was not balanced by array. The 9 had 3 small deletions in the P arm, and the Q arm had a 6.6 megabase deletion. The structure from the chromosome analysis is not evident from the banding pattern alone, and although the array shows multiple deletions, this does not give us any knowledge to the remaining portions of the rearranged segments. And even though the larger distal end of chromosome 3 is balanced, no part of either the abnormal 6 or abnormal 9 show a readily recognizable 3q. Our second patient was referred as a newborn for familial array studies to clarify previous chromosome results from a different lab. The chromosome analysis found that there was a 2q de novo deletion, and our array results show that there was a chromothripsis event that took place on 2q. The parental arrays were luckily ran concurrently, and we found that the deletions were actually on the paternal homolog. So as I stated earlier, there is a hypothesis that advanced paternal age is a contributing factor. However, this patient's father was only 29 at the time of the patient's birth. So we cannot rule out environmental exposure. But this patient was the only patient for whom we had both the father's age and array to show paternally derived deletions. So our third patient was referred for microarray at the age of 7 with no clinical information provided. Array showed a Chromothripsis event on the P arm of 17, including a terminal deletion. You see several duplications and several different copy number changes. The FISH was done and confirmed that we also found a duplicated terminus of chromosome 21. This duplication was actually on the abnormal 17 showed in the FISH results. And this case is not only an example of chromothripsis, but a phenomenon called telomere capture, where a deleted terminal end may borrow a terminus often from a different chromosome, resulting in a duplication of the borrowed region. And it's important to note this patient's father was 43 at the time of conception. So luckily, Chromothripsis has allowed us to simplify our array nomenclature. Instead of using this long way of writing these rearrangements, we now can use the shortened way with CTH designated as Chromothripsis. So my last patient was referred for chromosome analysis to rule out a possible mosaic trisomy 13. The patient did not exhibit phenotypically with presentations of trisomy 13. Our patient had intellectual impairment, developmental delay, speech delay, and several other presentations. The mother reported that the patient had a previous chrome report that showed mosaic trisomy 13. We interpreted the rearrangement as a duplicated region of 3Q. So, as you see, there is a very complex array going on right here. So this is a chromothriptic event with loss gain of normal copy numbers spanning about 16 million base pairs. You don't really see this complex nature in constitutional diseases. This is mostly seen in cancers. This is quite amazing how complex this is. And you would not think that all these different copy number changes would take place in 3Q. And based on the copy number, there are as many as 20 breaks involved in this chromosome. So chromosome analysis does help us understand structural rearrangements, even if the composition is not clear, but resolution can often be a problem. Microarray, however, can show the complexity of these rearrangements, even if the structure, but it cannot tell the structure of the rearrangements. Microarray should be considered for patients that have de novo mutations or unclear chromosome results. Although previous results show a strong bias towards advanced maternal age, we only have one patient that we know of that was over the age of 35. A few acknowledgements, thanks to the team at LabCorp and the laboratory team as well.